Hi, my partner Sofía and me are going to present an HIV-1 vaccine based on bacterium-like particles, elicits, and specific mucosal immune responses.
HIV-1 general release. HIV-1 is one of the two viruses that causes AIDS, which is the most advanced phase of the infection produced by HIV viruses. This virus is transmitted through direct contact with body fluids such as blood, semen, and vaginal fluids. The HIV-1 destroys CD4 lymphocytes and impairs the cell-mediated immunity, increasing the risk of infections and cancers. Retroviruses like this one are enveloped RNA viruses, and their mechanism of replication is via reverse transcription to produce DNA copies. HIV belongs to the Retroviridinae family, genus Lentivirus.
The envelope of the HIV consists basically in a lipid bilayer that was taken of a human cell during the process of new particle gemmation, forming a sphere with 100 to 120 nanometers diameter. Attached to this bilayer, the virus has some proteins of the host cell, and it also has M. This last one consists basically in a trimer formed by three molecules named GP120, and it is bound to another glycoprotein that is GP41. The GP120 has the ability of joining with a CD4 receptor of the host cell. This mechanism allows the entrance of the virus to the host cell.
Overall purpose: to develop an effective HIV vaccine based on bacterium-like particles bound to JP120 trimers.
So the first method that was used in the study is the SDS page and the Western blot. To perform the Western blot, it is necessary to first make a gel electrophoresis. The SDS page is the polyacrylamide gel electrophoresis, which is a technique that separates the proteins by their molecular weights. In the Western blot, the previous sample must be transferred to a filter where must have specific antibodies for the protein of the sample. Finally, it will show the affinity between the protein and the specific antibody. In the study, these techniques are used to separate the trimers, measure the purity of them, and evaluate how much the guinea pigs and the mice were sensitized after some weeks of administration of the trimers and the verification of the correct molecular weight. Next, the vaccine preparation. In the study, they had to bind the VLPs, bacterium-like particles, to a trimer. This last one consists in the glycoprotein or GP120 bound to a particle of MTQ in the C-terminal of the trimer and the molecule of proton in the N-terminal. Besides, in the assay, they also tested trimer without the molecule of proton to test the functionality of it.
The elicits test. This test is used to identify an antibody or an antigen bound to an antibody that had to be set to a solid surface to avoid them detection. Then, to show the reaction, antigen antibody is used as conjugated. It is composed by an antibody and an enzyme. Finally, the enzyme acts on the substrates and makes a color solution in which the intensity of the color is directly proportional to the quantity of antibodies or antigens present in the sample. In this study, this technique was used to identify the binding antibody antigen. In this case, they use a monoclonal antibody to identify the type of antibody in the serum samples.
The immunofluorescence assay. This assay was made with the purpose of visualizing the antibodies that can recognize specific antigens in a tissue. In the study, it was employed the direct assay, in which first is necessary to set the antigens onto the slide. Then the person must put the sample that is supposedly infected, and finally they add to the preparation a human anti-immunoglobulin, marked to show the reaction between the antigen and antibody. And in the previous techniques, this assay was used in this study to show the efficiency of the union between the created trimer and the antibody.
Now, talking about the results, we can see that in figure 1 there were built two types of trimer for the study. One of them contains proton in the N-terminal and the other one not. Also, we can see that these molecules are used to bind the trimer to the BLP, something that can be difficult in the trimer that we do not have the linker. In the C-terminal, the trimer has the MTQ, which is the mechanism to keep the three molecules of GP120 joined. Finally, the TPA was used as a secreted signal peptide. In figure B, there is a BM page that shows that the trimers with the MTQ behave as trimers, unlike the ones that do not have it, behaving as monomers and dimers, probably after a process of degradation. This figure can show the functionality of the MTQ as a mechanism to the conservation of the trimer.
We can see two essays of DNA page, each one corresponding with the two types of trimers made after the purification of the sample. And then we see that they have very similar molecular weight, but the one with the protein has even more correspondence between the different samples. This can prove that proteins with protein are pure because all the samples are very similar, which means that the probability that the sample has another species of proteins is lower. We can see that even in making two different assays, the molecular weight was correct. For this, we can compare the concentration of proteins in all the assays. Also, we can observe the similitude between the same trimer in the different assays, which help us concluding the same as the authors. The antigens were pure trimer after purification.
So in this figure, the investigators evaluated the bonding of the antigens to the BLPs. In the figure A, in this one, we can see that only the protein with the proton bound to the BLP. The photo shows that in the samples of the trim without the proton and the BLP alone that was used as a standard, we can see any concentration of the protein. This is because probably during the preparation of the assay, the other two samples separate themselves in their more basic structure. So the particle will migrate to a lower molecular weight, while in the lane of the trimer bound to a proton, we can see an organized behavior near to the 150 kilodaltons. In this figure, in the C, the investigators use as a standard the bovine serum albumin to compare the concentration of the proton bound to the bacterium-like particle. Finally, the result was very similar to the 0.25 milligrams per millimeter, which the authors convert into the 30 micrograms of antigen bound to the 1 milligrams of BLP. So the authors use these results to conclude that the proton is a good linker to the antigen. So for the discussion, we take into account what other authors said previously. So Paula et al. Said that early studies have demonstrated that BLPs are an excellent vector for inducing mucosal immune responses, and they have been applied in the design of many different vaccines, and that agrees with the study. Pegu et al. Suggests that subset of HIV-infected patients developed BNAs targeting the Env-spike or trimer. These antibodies can effectively neutralize most viral isolates, and that agrees with the study. Burton et al. Said that it has been reported that the mucosal immune system is interconnected, and that agrees with the study.
Conclusions. After all, investigation is essential in the medical field for an effective problem solving. In order to develop the solution for a problem, it is important to use all the tools at our core to have the best possible result. Molecular biology is the basis of the most tools that help us to confirm a diagnosis of HIV infection. To confirm or discard any diagnosis, many diagnostics tests use biology concepts. And understanding the concepts that molecular biology gives to us, we can develop and evaluate the effectiveness of a vaccine that will prevent an HIV infection. And the article biology concepts were used to understand the form and the membrane properties of the virus and his interaction with body cells, and that became the objective of the vaccine.